Hello everyone, I'm Nu Li, and today I'm excited to share our research on identifying potential inhibitors for Nipah virus matrix protein. This work was conducted under the mentorship of my PI, Dr. Carr, using an integrated computational approach. Our study combines QSAR modeling and molecular docking with advanced time dilation to find new ways to combat this deadly pathogen. Nipah virus is a massive public health threat with a staggering mortality rate that can reach over 75% in certain regions. As you can see from our data, outbreaks in India and Bangladesh have constantly shown very high death-to-risk ratio. While its current R0 is lower than COVID-19, any mutation that increases its transmissibility could be catastrophic. It is currently listed by the WHO as a priority disease, yet we still have no approved drug or vaccines. To accelerate drug discovery, we developed a multi-layered in silico workflow. We started with a library of 953 antiviral compounds from PubChem. These were screened through a QSAR model to predict activity, followed by molecular docking into the Nipah virus M protein structure. The best candidates were then filtered for safety using admit profiling before undergoing 200 nanosecond molecular dynamics simulation to assess their stability. Our QSAR model was built using 56 training compounds and provided statistically robust with an R-square of 0.68. We identified 7 key molecular descriptors that govern anti-Nipah activity. Specifically, descriptors like MDEL12 and EIG06AA showed the strongest positive impact on a molecule's predicted effectiveness, providing a clear roadmap for what makes a successful inhibitor in this system. From our initial screening, we narrowed the field to top 12 ligands. We then subjected these to rigorous animal testing. 5 molecules stood out with experimental safe profile, falling into toxicity class 5 and 6, which indicates low systematic toxicity. These molecules primarily interact with residues ARG191 and ASM152, which are critical anchors within the protein's binding pocket. When we move to long-term simulations, one compound, PubChem CID 6474310, clearly outperformed the result. It demonstrated the most stable binding behavior and the lowest structural drift over the entire 200 nanosecond run. Our MMGBSA energy analysis confirmed it as the strongest binder, meaning it fits into the target protein more effectively than any other candidates we tested. Metadynamics confirmed that this ligand significantly restricts the protein's movement, effectively locking it into a stable, inactive state. We identified this ligand as isoclonogenic acid A, a natural polyphenol. It is commercially available and have previously shown antiviral activity in other systems, making it as a highly possible candidate for experimental testing. In summary, our computational workflow successfully prioritized isoclonogenic acid A as a top lead for inhibiting the Nipah virus. Our next step involves moving from the computer to the lab for biochemical validation. Finally, Dr. Carr and I want to thank the administration of Hennings College of Science, Mathematics and Technology of Kean University for providing the research opportunity, resources, and research release time that makes this work possible. Thank you for your time.